Okay, so today in this video we will discuss about the photo respiration, which is called the C2 pathway. In this video we will discuss about the C2 cycle or C2 pathway. Okay, as you guys all know, in the introduction I told you guys that in this photo respiration we are having three organelles which are involved in this cycle. So first one we are having chloroplast, okay, which is involved. Second one we are having peroxisome, okay. Third one we are having mitochondria. Okay, so we were having a molecule that was ribulose bisphosphate, okay, which joined with the oxygen in the presence of the Rubisco enzyme, which was having a capability to join with in the process of oxygenation as well as the carboxylation. So here you can see that it is joined, this one product molecule is joined with the oxygen in the presence of the Rubisco enzyme and making a product that is phosphoglycerate, as well as the formation of the 3-PGA. Okay, so this is the formation which was like formed in C3 cycle. Okay, so this is different, which was the product of this in C3 cycle, but today we are talking about the in the presence of the oxygen. So we are having a product that is phosphoglycerate. Phosphoglycerate. Okay, so phosphoglycerate will convert into the glycolate in the chloroplast, and it will release out the phosphate, inorganic phosphate. So the phosphate is removed, so we are having a glycolate. So glycolate will, you can say that, move into the peroxisome organelle, and in peroxisome organelle the glycolate will convert into the glycine, which is a amino acid. Okay, and after, when the glycolate will convert into glycine, it will release the hydrogen peroxide. Okay, so this glycine will move into the mitochondria. Glycine, okay, this glycine will convert into the serine. When the glycine will convert into serine, there will be the releasing of the carbon dioxide in the mitochondria. As you guys all know, I told you guys that the carbon dioxide in the photorespiration, the carbon dioxide will release. Okay, so the carbon dioxide will release and NADPH will be formed. Okay, so NADPH, you can say that, you will say that the why, you can say that you said that there is no formation of any kind of a ATP, NADPH. So you can say that the NADPH has formed. So we can say that the answer of this question is that the NADPH is formed in mitochondria, but in Calvin cycle the NADPH was formed in the chloroplast. So that's why there was the use in chloroplast, but this is produced in mitochondria. So this is like the useful, this is not useful, this is wasteful. So there is like you can say that there is the wasteful, the NADPH is wasteful to produce in the mitochondria. If it is produced in chloroplast, so that like it matters that it will help in the Calvin cycle. Okay, so the serine will move into the peroxisome and it will convert into the glycerate. It will convert into the glycerate, and then glycerate will convert into the phosphoglycerate in chloroplast. With the help of the phosphate, the ATP will convert into the ADP, and the one phosphate will join with the glycerate. So we can call that as a phosphoglycerate. This phosphoglycerate will use to make the ribulose bisphosphate again and form this cycle again, for the formation of this cycle again. Okay, So this was the cycle which is in the photorespiration. So this was about the cycle. So this is the wasteful product where the oxygen is like absorbed and carbon dioxide is released. But as you guys all know, we were having a useful process, that was Calvin cycle. In Calvin cycle, we were having a carbon, in the use of carbon dioxide, the glucose was formed and oxygen was releasing. But here the oxygen is releasing and not oxygen, here the carbon dioxide is releasing and oxygen is to be used. So there is no formation of any kind of a product, there is the consuming of the ATP. So this is a useful product just because in this the ATP is consumed. Okay, so this was about the photorespiration. In the next video we will move toward the C4 pathway. Okay.